Hi everybody, hello. So we are back in the lab. So here is this awesome laser lab we got to see yesterday, but now it's all up and running and I got some numbers. So hello everybody, how is everybody? I hope everyone's well. I'm in the lab today. This is, hey Victoria. So today I'm actually in not the lab I used to work in, but it's right next door. And if anyone remembers from quite some time ago, hello, a very special friend of mine, Alba, made a cameo. This was way back, I think pre-Android times. Hey Hal, nice to see you. Hi mommy. So we are, I'm passing through and she's so graciously let me come in and tour again since I've graduated now. I don't work in a lab anymore, not this type of lab, research. So great, yes, get your laser ready. Hey doc. So we are here, we took a little tour of this lab yesterday, so used my block solution. Oh, I don't know if I've been running around, so I hope that worked. That's awesome. Hey Ian, and after looking at all this kind of cool stuff, we can go over that briefly again, and we've got the microscope running so I can show you some of these really cool images they get, that they use these lasers, these actual pulses of lasers. Hey Orion, hey everybody. Ian the neuroscientist, excellent. I don't know if Ian's here. If he stopped by, I'm gonna finally be able to catch his shows when I'm not traveling anymore, but he is fantastic. So at underscore anthropoid, I believe, he's a neuroscientist, he's a grad student, and he's in a lab all the time, so he has really cool scopes. So, excellent. Oh, thanks, Ian. Alright, yeah, so without further ado, I'll show you the lab again, what we looked at yesterday. I'll kind of briefly go through it, ask any questions you want. Neuroscientist, Ian's great. He's got so much stuff in his brain. He's so informative. Ask him questions, definitely. What are we going to look at? Great question. Probably Orion. We are going to look at the lasers first. Nice to see you as well, Laura. We'll look at some of the images too. So ask questions as you want. I'll go through kind of the process of what's going on. And we have worms we can look at. We've got some cheese and some other stuff we can look at too. So he is great. All right, laser ready. Excellent. So, I should also say hello again. If you're new, my name's Julia. I'm at Fine Woman here. I do a scope show every weekday, Monday through Friday. Usually it's at 9:30 on the West Coast in the morning, and that's 12:30 on the East Coast. But since we're doing lab tours today and getting access and getting in here, or going around group meetings and things like that, we had to change the schedule. But please join me if you're around. If you don't know me, come back every day. So tomorrow we'll do a show. It'll probably be an AMA, but I'll be back in Dallas and we'll be doing demos and all sorts of really cool at-home science stuff. So I like doing that, and I have wonderful audience here. So feel free, always ask me questions. Please interrupt me. If I don't explain something clearly, let me know. I'll try to find another way. You can find me at finewoman.com/periscope. So we'll be using Swiss equipment. Yes. Yes. I don't know if it's maybe German though. Don't tell the Swiss. Thanks, Ian. Alright, so here we go. This is the system. Hey Linda, welcome. Thanks so much. Oh, lasers, it's hard not to get excited about lasers. So I'll go around here. There's two microscopes in here actually. Here's one, and here's the other. This is the one we're going to be using. I'm in Dallas, Orion. And then these are some of the images I'm going to show you that were maybe sort of prepared ahead of time, but they were taken recently. Okay, so we'll go around, and I think also people wanted to see the holes in the curtains from yesterday, so I'll show those briefly. So these lasers can get powerful enough, so you can see here some of these holes in the actual curtain, there we go, where the laser has punched through. So not so careful there, but that's just that's to show you how powerful some of these lasers can get. That was some of our diagrams from yesterday. That's awesome. So this is actually we have two lasers here. Well, actually technically I guess it's three, but this one first, there's actually a beam coming out right there. And now you can't see it. Oh great, no, I'm glad it's kinetic. So you can't see it actually because it's 1064 nanometers is the wavelength of the light, so it's 1064, they kind of abbreviate it, and that is out of the visible spectrum, so we can't see it with our eye, and it seems to be outside the spectrum of the camera. So that's a little trick actually if you want to see if your TV remote is working and it's still one of those infrared signals. You can point it in your phone's camera here and you can see it blink, which is kind of neat because it does pick up some of the stuff that our eye doesn't pick up. but this is beyond even that, so you can't see that one. That beam comes and hits this string of mirrors, actually, comes all the way back around here, and then comes this way, and we go in there. So yeah, this is a pretty cool setup. So that's one laser, so that's laser one. The other one there, we have, that's that green color. So that green color is 532 nanometers. So again, your hair is about, let's see, 10 to 100 times that wavelength of light. So we don't actually make holograms in this lab, but there is another one on campus that plays with that stuff for sure. So that laser, actually what we call pump, which is to stimulate some of the lasing processes in this big old thing. And this big old thing is really cool. It's called an OPO, an Optical Parametric Oscillator. As I was reminded, because I'd worked with optical parametric amplifiers, which would be OPAs, and they essentially do a similar thing, but they amplify in power what you're sending through. So, oh, Linda, thank you so much for telling me. And also, if you have, definitely follow Linda right there who commented. She's fantastic. She does amazing knitting and teaching scopes. So, and she's got a hat that she made us, or Jason and me, and Jason's going to show it off, which is awesome. Okay, so what this OPO is, this Optical Parametric Oscillator, the reason it's called that is because it takes this optical oscillation, and oscillators are another word for laser that used a lot because you're oscillating. It's taking these oscillations of, an oscillation is just a wiggle, right? So if you have a wiggle, that's what light's doing, and so it's capturing that oscillation, building up in power. So this optical parametric oscillator, what that does is it actually takes this light and it uses that to do something called pumping. So I've talked a little bit in my older laser scopes, and again, feel free, please ask me questions if you have them, or if you have any, if you want me to show you something that you can't see or I've passed over it too quickly, always let me know that. So thank you ahead of time, I suppose. All right, so the pumping process, that pumping, that laser comes in and pumps, which is exciting these atoms or the material in the laser that is going to give you the light that you want. You want a very specific color. The endpoint of the first laser, well, it's a little complicated because we actually combine the two colors, and I believe, let's see if I get it right, so we combine here. That's a beam splitter. So the two colors of light are actually combined together in the same path here, and then they both come here, well, a little bit more complicated here, and then they go into here, which goes into the microscope. So that's the endpoint. The endpoint is the microscope, and then just like any other microscope, you'll see there's some slides, you know, little lenses there and objectives and then slides. So that's essentially the endpoint of the laser. It comes up through this tube and into that the objective that you pick. So we are doing a lab tour here. All right, so this is that is the endpoint because that's the microscope where the light from these lasers is going to combine and give you these really cool images. So, oh sure, so you won't be able to see the light reflecting because again this is yes it is, Tatiana. So this is 1064, the light that comes out of that laser. There is currently light coming out of there, and that is 1064, so we can't see it with the naked eye because it's outside of the visible spectrum, infrared, exactly right. So that is, that's where that light comes out, and the mirror there that it bounces off of is at the angle. So remember, reflection, if it comes in at one angle on a mirror, it's going to bounce off at the very same angle. So great question, and I will absolutely get into why we're shining the lasers, because there's a few different ways you can do that. So I'll try to do a very quick explanation that gets the main points and not lose too many people here. But essentially what you're doing with those lasers is you are pinpointing a certain molecule by exciting it in a very specific way and then having it light up for you. And so we're going to do that with the fats and the lipids. Alright, so this optical parametric oscillator, what is really cool about this is because of the way that the light, that green light, pumps the light in here. Oh, thanks, Tatiana. Let's see. Some sensitive paper? Yeah, the infrared paper. Huh. Okay, I'll take a look here in just a second, because if we can see that, that would be cool. Yeah. So Tatiana would absolutely know too. Tatiana's a fellow physicist. So the OPO, what that allows you to do is tune. So we've talked a little bit about lasers before, where you get a laser light that comes out and it's going to be one specific color if it's always on, right? If you have pulses, it's a little different. But great question. So there's a lot because first of all, well, it depends on the system, but part of it is because you want the path to go exactly, you want it to be in a very orderly path so that you can control the distances that these things are traveling. The main reason for these systems is that what comes out of this OPO, which is going to be a tunable central color, so a main color, and tunable means you can kind of, by the mirrors and the prisms and things that are inside, you can pick what color comes out of that, and it comes in pulses. So it comes in like little shots of light, and those pulses give you, it's like a shutter speed on a camera. It allows you to, when you have two pulses, you can overlap the pulses or not overlap the pulses. Now, it can't do this kind of thing with electronics, right? Because if you do, here, I'll do a quick hello too while I'm here. So if you can't do these things with electronics necessarily because electronics are too slow. So when you shut on a switch on and off, colors here are green and infrared, which we can't see, and also mostly red colors. So hey, Jackie. So in electronics, if you want to switch on and off something like a laser to have a shutter speed, then you are restrained by how quickly those electrons can move into the in your circuit. And that's actually really slow when you compare how quickly light moves and how quickly light reacts. So the only way to get a shutter speed that's fast enough to see some of these things, and I'm talking like, so we've talked femtoseconds, picoseconds, these are millions of seconds, so fractions and fractions. So to get those really, really small fractions, you actually need light to interfere with itself, because electronics is just too darn slow. So when you get light interfering with itself, that's the thing that's going to be fast enough to give you a shutter speed. And when you want to see things like molecules moving around, it's really cool. When you want to see molecules moving around, or you want to be very specific on which molecules in space or which molecules in time that you want to see, then you need to have those pulses overlapped very carefully. So what happens is, and here I can even I'll draw a little picture. So what happens is, if you want a pulse of light, let's say you have one pulse and then you overlap it with another pulse. So you can kind of see the drawing there. I had something on the back of that paper. That's terrible. Here we go. So, sorry. I'm back. So we have, if we have a pulse of light here and another pulse of light that comes later. So there are my pulses of light. So if this is time, right? Interference, yeah, it is, it's very clever. So what happens is, if this is a pulse, right? This is light, and so it's traveling at the speed of light. And if it's traveling at the speed of light, that's really stinking fast, right? And you have two of them, you want to match them up exactly where you tell them, right? If you want that shutter to open or close, then you're going to be overlapping these either right on top of each other or a little bit far apart, and that'll tell you like partially closing or partially opening the shutter based on how much they overlap. So by doing that, you have to be very, very, very precisely controlled in the distance that these beams are traveling. So if you have two lasers, you've got two pulses coming out, and you want those to overlap on top of each other, they have to travel exactly the same distance. And exactly means like you're talking about the wavelength of light. So that's, again, these are hundreds, tens, hundreds, thousands of times smaller than the width of a human hair. So you can't even see these with your eyes. So to precisely control those distances, the angles, you want to make sure one pulse, right, this is in 3 dimensions, so these pictures are nice, but in reality you have a beam of light, and so you don't want one coming in at an angle and one going straight on. You want those to overlap really nicely. Sometimes you do want them coming in at an angle because the direction, absolute light is a vector, so those those intimidating words in math, sometimes I don't even, you use vector and maybe without having to kind of really move these mirrors around and stuff, it's hard to appreciate the power of a vector, because that just means it's got direction to it, and that direction matters. And so when you have these pulses of light that you want to map on top of each other perfectly, then the direction is going to matter because it's got a shape to it, it's got geometry to it, and so something like a wave, they call it wave fronts, that's that front of that beam, you want those to line up in a very particular way. And so you can have, you can excite molecules and you can excite different materials and atoms based on the directions that those are coming as well. And so that, because it's a momentum thing, direction is momentum, so you absorb, it's not just energy, it's also momentum. So, any questions? Follow me so far. So if you have any questions, please ask. But that was from Ian's question. He was asking, why do we have to have so many mirrors and have them so precisely controlled? That's a big part of it, especially in pulsed laser systems. So, oh, thank you, I'm glad you do. So, yeah, great. So that's why there's so many mirrors, and that's why you'll see, I'll show you here also. That's why here you'll see this these two that are kind of facing each other, and we were looking at corner cubes yesterday. People were asking me, you know, what are the uses of oh, thanks, Sky. What are the uses of corner cubes? Well, that's one of them. So if you want to change the path length of something, of the beam, for instance, so a beam would come in here and then hit this mirror and come out here, and oh, thanks. Thank you very much. And yeah, if there's something like I speak quickly a lot, I know. So questions are always welcome. But thank you if people are starting to get this. So thank you. Yeah, definitely more women, more people in science also. The on button. So the on button is surprisingly simple, actually, Orion. It's a good question. So a lot of these are also controlled with computers and stuff. So yeah, I'm sorry, yeah, I was getting off track. So these are at an angle because a beam coming in here is going to be parallel to the beam going out here. So if all you have to do is move this back and forth, that changes nothing else but the path length, right? The distance that pulse, right? So this pulse is here and it's coming here. And if you have another pulse coming in and you want them to meet here, well, you might be off by what a micron. A micron is you know, half the width of your hair, or a tenth of the width of your hair. Then you have to be able to control, and that's what this stage does. This moves very, very precisely so that you can not change the direction of that beam. You don't have to change where that wave front, if it's pointing this way or if it's pointing that way, but you can just add a little bit of length to that full path. So a demo. So we're going to look at pictures here in a second. And let's see, I'm going to do this very, very gently because... No, I won't do it, because sometimes you can move these by hand and you can kind of see the move, but I don't want to. This is not my setup, and the worst thing you could do is move something and not that somebody else put together, so I won't do that. Yes, we're going to look at pictures of really cool stuff, so here's a preview. These are lots of pictures that we got set up here that have been taken with this microscope using these lasers. So these are worms, actually. These are called, I believe, elegans, which is a great name for a worm, I believe. So look at that just a quick second. Just go finish through the lasers. So this tunable color that comes out here, that is what comes, and then I was saying here, they recombine on this mirror. I'll give you a little bit of perspective. I thought you turned on. Oh yeah, so not all the light we're going to be able to see. So that's the 1064, which we can't see. 1064 is the nanometers. That's the wavelength of the light beyond what our eyes are sensitive to. Yeah, absolutely, Orion. So these lasers, this is a very specific type of microscope that uses the lasers in order to see a very specific type of molecule. And then, so that light comes out here, sorry, that light comes out and goes through here, and then we recombine with the light that comes out here. It goes through this path, and then we come and we combine them again here. So that's our two pulses that are going to be combined, and then we send them around, sorry, I'm looking through the camera, but we send them around here, and then it comes into this box here. To a large degree, and it is. To a large degree, it's not. So something, for instance, like this, I'd have to align these kind of things all the time. And so what you do is you can take like a piece of string or a ruler, and you measure the distance to get close. So hey, so you get pretty close because you know you don't want to be a centimeter off, because then you're way off. Because how much distance does a pulse of light cover in a centimeter? Or sorry, how much time does it take for a pulse of light to travel a centimeter? Like, you know, infinitesimal. It's a really, really small amount of time. So you have to be so you get pretty close, and then the angle is what's really the pain in the neck to align. So these systems, it is such a pain in the neck. So you see these two knobs, and they're on every mount. Sometimes they have 3 knobs. but what those knobs do is they control the mirror tilting this way and tilting this way. So hopefully that's kind of clear. So you're gonna have a mirror here, and you want to tilt it this way, and you want to tilt it this way. So, hi Scarlett. So what is the advantage of laser microscopes? Ah, great question. So the advantage of this is the resolution mainly. So mostly, that's true, that's a great idea. Thank you. Let's see, Tatiana, if I can have, is this one working? I think this is working. So let's see if we've got a beam. Ah, okay. Yes, all right. So here is the power meter. So I'm out of the way, and we're dropping. Can I catch it? There we go. Nope, no beam there. So she may have blocked some. Oh, I think she put a beam block in. That's why. So let's see if I can. And if it's tuned 1 milliwatt at 868. Okay, so this one is not sensitive to the light that we're looking at, but we'll see if it picks up something. Great idea though, Tatiana. So let's see if we can. Nope. All right. So this power meter. So again, thank you, Ryan. So again, oh, and this is also this. See, this is why it's good to have more than one person in the lab, because sometimes you use a power meter that's not on instead of the power meter that is on. And you see random fluctuations of just regular light. So that was not the one we wanted. Let's see if we can turn this on. Nope, not on. Okay, so I don't want to mess, again, this is not my setup, but we can actually see, if you want to trust this, this is actually telling us the colors. So that's the tunable aspect. You can tune the colors, and then you have the two different colors of the signal that are coming in. But apparently what's coming out of this is about 1 watt. I did not know this. So it's definitely not, yeah, it's a little bit of trial and error, but that, Ian, comes kind of at the, that's the last step, is those final little tunings. So these mirrors that go back and forth and left and right, that is to do that really, really tiny, precise measurement. And then what you do is you have things like this, and you see those drawings on them. So, and the burn spot, actually. So that is because you put this in the way, and you usually have something called an IR viewer. So this is called an IR viewer, and let me see if I can see anything. Ah, very cool. Okay, let me see if I can show you this. So let's see how this works, if at all. This may not work very well. There we go. Okay, so you see those, that's a green dot, but you can kind of see, kind of sort of there. So it's a dirty lens, but you can kind of see those dots that are coming, that are next to yeah, you totally see, awesome. So this is an IR viewer, the infrared viewer, so this is sensitive. This is like the night vision goggles. It's actually pretty much the same. And so what you're seeing is through these viewer, it's picking up the light that you couldn't see next to those mirrors. So you have to make sure the light's on, and so you can do that with these cards. So you look through the IR viewer, you do a little dot as to where your beam is, and then you kind of go back and forth and make sure it's going in the line that you want it to go to, and the height's the same and all that, and that's how you, that's when you do that final type of adjustment there. And sometimes it's trial and error for like the distance here. Sometimes you just have to take a measurement, get close, and move it around and see how it goes. But mostly you do the best you can to preparation, kind of think before you do things and try to organize it. Because also, you know, this space, it looks like a lot, but if you put a mirror here and then that's directly in the way of a mirror you need to touch a lot, then that's the trial and error part for sure. Okay, so without further ado, we'll come over here. I'll say another quick hi, just in case anyone has any last questions. It's totally sick, bro. Totally sick. This laser is like crazy. So, oh, thanks, Parker. Nice to see you here. So this is the final bit. Final bit here. So we come up, and then these pulses have been combined by the time they come through here. And so this is, you can see these are apertures just like in a camera. So all that does is kind of block. It actually gives you a center point to keep the beam in the right direction. Oh, good question, Ian. you need a lot of patience. You are so, yeah. So honestly, it depends how well you know the system. So something like Alba, she's worked on this system for so long that it would probably, if something got messed up, it would probably take her a number of hours. For instance, like the laser that I was working with in grad school got misaligned, and that had tiny little mirrors on it, and it took me months to get it realigned. But then the next time I do it, Aces Hella, exactly. The next time I did it, it took me 2 weeks, and then the last time I had to do it, which I shouldn't have had to do it all, I guess, but as much as I did, but it took me, you know, a day. So it depends. It really depends on the system. It depends on how messed up it is. It depends on if you know what the problem is, that kind of thing. So once those beams come together, and you know, this is kind of why a graduate school or these degrees take like 6 years. So I was talking about molecules, but we're going to talk about them again. So this is an iris. Oh, awesome. Awesome. Thank you for inviting your friends, everybody. I really appreciate that. So once we get into this cool box here, then this has an aperture in it, and then it goes these combined pulses. So remember my beautiful picture here. Then these are 2 pulses of lights. So some of them are, Martin, yeah. Yeah, it is. That's a very good point, Barbara. Some of them are, and it depends. So some of those stages, for instance, that stage right there, that's a manual stage, but they do have motorized stages. So motorized stages, and actually when you want to do really precise measurements with motorized stages, then precise measurements, motorized stage is the way to go, because then you have very, very fine control. So, oh, very soon, I think I'm going to wrap up here. I think my battery's also dying, so I'll wrap up in about 5-10 minutes, hopefully. So, where would the, yeah, exactly, Tatiana. So, you got to do everything by hand. So, once we come through here, we have these 2 pulses, and what we want, so when the pulses align, and that time, again, this is like a shutter speed for some of these reactions, or some of these interactions. So what you'd want is you'd have a pulse here, and then you move those, those 2 corner mirrors there, and then what you do is you can get this other pulse to line up perfectly here, and you, and then what you'll do is during a measurement sometimes you'll move this so that you can tell the difference between a measurement made here and a measurement made here, and that has some information in it. So I don't know who, I actually don't, maybe I know them by a different name, but I'm sorry, I don't know who but thank you for joining. So now to the pictures. So these lasers that are going to combine here, we're going to come up through that microscope objective, and so a slide is typically going to go in there, and it's going to be, there's a camera up here, and so the camera is going to pick up what goes up this objective, and so the lasers are coming up through that bottom objective there. And what that does is, using those 2 very specific colors, those specific colors are going to excite specific molecules, and in this case lipids, these fats, and these are all fats. So this is the fat on this worm. And these light up because those lasers are very specifically exciting the vibration of this type of molecule. So based on the structure of this type of molecule, those pulses of light, oh, thank you, Scarlett, thank you, I appreciate it. So based on those pulses of light, those, and exciting that type of molecule, you can get a very, very precise image, unlike, for instance, a light microscope. Now, depending on what you're looking at, so Ian, for instance, he uses, I think, a fluorescence microscope. It's a little different. The resolution's not quite as good, but he can look at a larger range of things. So it's a bit, it is kind of, it's sort of, it's more of 2 microwaves combined in order to look at something even better. But yeah, it's pretty close. So here we've got, where did she show me? So this is, Alba took this picture. So these are the eggs that the worm stores, and these are a nice calibration kind of type of thing to look at with these microscopes, because that fat that they're looking at, these lipids, it's about 2:30 in the afternoon, I think. Those light up like a Christmas tree, as you can see. They're really reactive to this very this nice energy, and so you can see all sorts of stuff. And apparently, let's see, where else are, so I think these are also some eggs in the worm. This is the mouth of the worm. This is the tail of the worm. So that's pretty cool. So you can do these nice calibration types of things, and then those again are the eggs apparently. Yeah, exactly, and that's a good point actually. So it's a joke, it's funny, but that's really what's partly interesting, because they can feed these worms differently, and based on what they feed them and how. So what Alba studies a lot is cholesterol and the different types of cholesterol, and so based on what they feed these worms, they can see how those fats, those lipids, will rearrange in the worm, and from there I don't think I could tell you anymore, but that's not my expertise. But the other things that we hate equals block. I'm not sure what that means. Does it damage the tissue of the worms? No. So these worms are, I'm assuming they're dead. So fat worms, notice they do. So these worms, I'm assuming, are dead when they took the image. In fact, actually, I'm certain they are, because they would move around, which would give you a terrible image. But the actual effect on the worms, I don't know. I can ask Alba. I don't want to give you the wrong answer. So yeah, definitely dead. Sorry, it took me a second to realize that they're definitely dead. So this, just for some fun images, again, this is all these fats, and the reason they can look at these fats is because they're looking at a very specific stretch of oscillation in the stretch. How quick is the capture? Oh, that's a good question. So these are captured quite quickly. So I think the pulse length is in picoseconds. So again, that's millionths of millionths of a millionth of a second. CH bond, thank you. So it's a CH bond and a CC bond, and it's actually a combination, the right combination of a couple of those oscillations. So it's this kind of stretching of the molecule. And no, Scarlett, they're not. These are called what did she say? Elegance worms, I believe. So Rev, hi Rev, so nice to see you. So let's see. This is ricotta cheese. So again, it's very similar to these fats. So cheese obviously has quite a bit of fat, and that those oscillations, those molecular oscillations, are what kind of light up here to varying degrees. Yes. So can you also? So Ian, I think yeah, the color. So you can't change the color if you're talking about just a laser in air. You can't. Yeah, you can change the rep rate, and the rep rate is how many pulses per second come out. And I don't know that, Scarlett. There's lots of arguments on it, though. The rep rate is how fast those pulses come out. Now, the color is the energy, essentially. So if you're tuning the wavelength, you're tuning the color as well. So the wavelength in air, again, then you're tuning. And again, when I say tuning, it's within a range, so it's not any color you want, and you need a different laser, a different setup for a different range of colors. But the colors that come out, or the wavelengths, yes, you can tune them and change them, and then that changes the energy to pick a different type of stretch or different type of molecule to look at. So, oh, thank you so much. Yeah, so we're going to wrap up here shortly. I'll just show some more of these pictures or answer any last questions that people have, so we can look. Oh, I think that was probably Linda, I think. So here, this is an onion skin. So that's pretty cool. And for scale, I actually don't know. Do you know the scale of this, approximately?
100 micron by 100 micron.
Oh, thank you. Okay, so these are 100 microns. So that's about, what? About the width of your hair, maybe twice the width of your hair by the width of your hair. So that's how big these images are. So this is an onion skin, where if you took a little tight piece of onion, the size of a width of your hair by a width of your hair. So these are, these are kind of some of those stored. Obviously, onion doesn't have a lot of fat, but you can see some of the little resolution here in the structure and how those get stored. So, ooh, very cool. Ah, the app is evolving. So let's see what else I can show you. Any last questions? I guess I'll just show you some more of these. Oh, these are more of the worms. These are eggs. Those are eggs inside the worm. Optics, that's true. Let's see. Oh, these are some of the cells that Alba studies, actually. So these are apparently, oh, I am? Wow, Rev, thanks. I'm 29. So these are the nuclei. So the nuclei doesn't have any of these lipids in it, but they look at these lipids. Is this info? I can't tell. So it's, oh, the image itself is just a little bit blurry. So these are the lipids that she's looking at, and the nucleus of the cell is here, and it is a little hard to tell. Maybe that's a little better. You can see some of these, the very, very mild red here, and then these are really what light up, and then the nucleus doesn't have any of that inside, so they can kind of tell what part of the cell they're looking at. And I think the last one, oh, this was a nice little flower. I think we were doing some demos yesterday, so somebody got a flower they picked outside, and then they took an image there. And so that's using this microscope, with that camera, and then that's from the lasers that come out here and mix together and come through here and then excite these very special molecular stretches that they tune to in the vibrations. And then they get this image. XP, I'm not sure what you, I'm 29, Scarlett. I'm not sure what XP means. Windows XP? Then yeah, most of the computers are still, at least I did, we were trying to hold on to XP for as long as we could. So, hey Mark, nice to see you. Yeah, so I'm sorry, wrapping up now, but any last questions about the laser, the system, the microscope I can answer? I'll see if I can ask Alba some of the questions about more questions about her cells. She's not feeling well, so didn't want to drag her on here, so, well, thank you, Scarlett. Do you know the resolution of the images? Yeah, oh, good question. So I think, so if this is 100 micrometers, so that's 100, so a micron is a millionth of a meter, and so this would be 10,000th of a meter by 10,000th of a meter, and I think this is 1024 pixels by 1024 pixels, I believe. And that's, so you can, we can do the math, I suppose, or we can do it offhand, but that's going to be 1024 times 1024, and then you can divide that number by 100 times 100 micrometers and figure out what the pixel per pixel resolution is. But great question, thank you for asking. So anything else? I will try. I thought I was going to get into my friend's lab. He does some really cool studies with magnetism. I'll see if I can catch him, but he was running around today, so I promised to. I'll see if I can get at least a quick look at his. But sure. How many images of individual? Oh, well, thanks, Tatiana. Bye, Victoria. Individual molecules, that's going to be like single molecules and resolving like unless it's a really big molecule like a protein or even DNA. If it's like just a very small molecule, a few atoms, of course, Barbara, that's going to need something called a scanning tunneling microscope, which is usually at really cold temperatures, and they have one of those next door actually. So if they're around, I'll see if I can sneak in there too. But well, thank you very much. Thank you everybody for stopping by. Thank you for all the lovely hearts and the support, and I appreciate it. And so I guess I'm close to 1 million, which is incredible. But thank you everybody. So I've been on for a while, and Rev has too. Love Rev, he's so sweet. So of course, thank you, Ian. Thanks, Orion. Bye, Tatiana. Bye. I do one every day, Scarlett, every weekday, and my schedule's in the profile. So you know, Eastern, etc. So. Thank you everybody. Bye, Orion. Have a wonderful, wonderful day. Happy science, and Periscope, down Periscope, I should say. Bye, everybody.